Another agent that got some discussion and may have a new biomarker to treat is dabrafenib as a RAF inhibitor and the BRAF V600E mutation. What did you think of this as a potential marker? Is this something that you think could be used in the clinic? Are we going to reach a problem where these are too rare to test for, or how are we going to move forward with these?
Yeah, I actually have a BRAF trial open, and it's taken a very long time to get our first patient. Just for that reason, this occurs in about 1% in our population of patients that we've been testing. And because we have a trial, we've tested every single patient, and that's about what we've gotten in terms of hit rates. So this, of course, is a multi-institution trial, but I think these trials may still struggle with accrual of these patients. The results, I think, were certainly intriguing. This is a population of patients that, it's not the typical mutation population of never smokers. Many of these patients are former smokers who sometimes can have be a bit more of a challenge with later lines of therapy. And it looks very intriguing, I would say. I think it may be a challenge to continue. Some of the toxicities may be a challenge. And I think in melanoma, we're seeing in this population of patients that the melanoma folks are moving more towards combination therapies with, say, a BRAF inhibitor and a MEK inhibitor. And I think that we may do that. I think it's just going to take us so long to get through the BRAF, and then doing subsequent trials may be challenging.
Do you think that we might have an easier time? I mean, certainly one institution with a 1% hit rate is going to be a real challenge. But Nate, I know you had commented on next generation sequencing, and we may be moving quickly in the next couple of years towards multiplex testing for a battery of things, where a much broader population of patients across the country is found to have rare mutations like ROS1 and BRAF V600E and a few others that would otherwise be infeasible to test, HER2, et cetera. So are you optimistic that we can do trials for these 1% populations, or is that still a very elusive concept?
Well, I think the reality is we absolutely can do it, even in rare 1% drivers. I mean, there are more than 150,000 people with lung cancer in the United States every year, and that's plenty of patients to find these patients and do trials. The problem is we're going to have to rethink the way we are doing the trials, because right now an investigator has a relationship with a company, they get a drug, they say, hey, I'll use my 500 patients a year that I see and I'll put them on the trial. If it's something like EGFR mutation where you only have 10% of your patients who have it, you say, well, maybe I'll call my friends and we'll have 3 or 4 different sites, and between all of us we'll be able to find enough. Well, now you have to call 150 friends if you want to do that. And practically speaking, we're going to need to use something like either the cooperative group mechanism, where we already have a mechanism in place to open trials at hundreds of sites, or something outside of the cooperative groups. The Biomarkers France trial showed that on a country-wide level, you can do this kind of testing and identify these patients. And the next step would have just been to open your BRAF trial in France at these sites, and these patients could have been directed to the trial, and in a couple of years, you'd have known whether the drug worked.
I actually think that there could be a use for online groups. You have a lot of patients with ALK rearrangements and ROS1 rearrangements, and they're finding each other online and GRACE and other communities, and they share all this information about novel agents. And even though they're not that many of them, they're all finding each other, self-aggregating, and sharing information about new drug being tested at whatever place. And, if you have agents that have a promise of response rates beyond 50% and some of these very long-lasting, this motivates people to even travel for them. We'd love to have our trials available within ready driving distance, but if you have a really promising treatment and you've got a 1% mutation, the value proposition might be there to go where the trial is.